if you think of sugars, you think of cakes, you think of sugar that you sprinkle into your tea, or you think of the sweet substances in honey. But we as scientists think of sugars as much more complex molecules that have very diverse function in life. All cell surfaces are covered with sugars, regardless of whether these are cells in our body or in small bacteria or in plants. All of these cell surfaces carry sugars. And the cells use these sugars to communicate with the outside world.
Sabina Flitch and her colleagues are developing a device that mimics this property of cell surfaces. They use a gold plate coated in different sugars. When the plate is placed in a solution containing an unknown species of bacteria, the bacteria stick to the sugars. But which sugars they stick to depends upon the species of bacteria. Thus, the process can be used for diagnosing infections.
In this row, I use one sugar, which is glucose. And as I'm finished with this row now, for the next one I can use a different sugar, for example, mannose. So then in the end, we will have several different sugars on the same gold plate that can be tested with bacteria. So what I've done now is I have different sugars on the different rows of the gold plate. I left it overnight so the sugars could bind to this gold plate and washed it. Sugars are still on. And now I could apply a solution with bacteria that I don't know which bacteria they are at the moment to this gold plate. And the bacteria have preferences for different sugars. So they just bind to those rows with sugars that they prefer. And now we can analyze the different rows and see where we have bacteria binding to the different sugars. So this is the result of the analysis of the gold plate. We see that on the row 2, the mannose spot, where we have the sugar that bacteria, E. coli bacteria bind to, we see that there are spikes in here. And we can see that we get signals. On the other rows, we don't see any signals at all. So now we know that it's E. coli that have bound to the gold plate.
So these gold surfaces could then be used as tools to detect harmful bacteria, which you might find in hospitals or in food sources.
Also at Manchester is Simon Turner. He is developing means by which cellulose can be efficiently extracted from cell walls of plants for use as biofuel.
So this is a crop that you might one day grow for biomass. It's actually a small poplar tree which has been actually modified. And there are two things that we really want to do. We want to generate a lot more of this wall material. So we want these trees to grow faster and generate more wood. And the second thing we want to do is to alter the composition of the walls, because typically the walls of this plant are going to be about 50% cellulose. And it's those other components in the wall which are going to limit the digestibility of that cellulose. Hopefully these plants are going to be more easily degradable into sugar. We can use that sugar, convert it into biofuels like ethanol, and we can burn that in our car. Now the great idea about this is that we're going to use biomass. We're not using food, so we're not competing with food. We can also use the residues from crops. So if this process is really efficient, then it's the carbon impact of that process should be really small.
At Imperial College, Stuart Haslam is investigating the role of sugars in human fertilisation.
So the human egg cell is covered with a layer of sugars on its surface. And what our laboratory did was characterise which sugars on the surface so that we can identify the specific sugars which are recognised by the human sperm to initiate the fertilisation process. So now that we know the actual sugars and the actual molecules which are present on the egg, we can then start to both inhibit fertilisation and hopefully develop new, more effective contraceptive molecules, but equally we can start coming up with new experimental strategies to improve fertility rates and improve the success rate of in vitro fertilisation.
So there's more to sugars than just sweetness in your tea. We've shown you 3 examples, how sugars can help with human fertility, how sugars can be used to develop diagnostic tools to detect harmful bacteria in hospitals, and also how sugars can be used to make biofuels from waste materials from plants. The future of sugars is sweet.